My name's Diana, and I'm a PhD candidate at Brandeis in the Griffith Lab, and I'll talk a little bit about my path in science so far and what I'm currently working on. So I'm from Michigan, from the Metro Detroit area, but my grandparents live in northern Michigan on Lake Huron, and I spent so much of my childhood there. So of course I have to make sure to shout out the Great Lakes. Um, for me personally, ever since I was young, I knew I was really interested in the brain and how it works and just, the human mind. And I did like science. I did Science Olympiad in middle school. And in high school, although I was still really interested in my science classes, I wasn't necessarily doing science specific extracurriculars since I was involved in a lot of different activities, music and sports, and volunteering. But since I was interested in science and the brain, I had a lot of people telling me that I should go to medical school and become a physician. And I wasn't entirely sure what I wanted to do, but that seemed like maybe the closest that I could get to what I wanted, but I knew it wasn't quite what I was looking for. So I went to college, mostly just knowing that I wanted to go into neuroscience, but not entirely sure of whether that meant I had to become a doctor. I just knew that I would get to devote all of my time to studying the thing that I had always been interested in. So coming out of college, or out of high school, I thought that I wanted to go to a small liberal arts college, and I also had to pay for college myself. So I went to a small liberal arts college on the west side of Michigan that I had gotten really good scholarships for, and I felt like it was a good fit for me. And I knew that I was interested in science, so I had applied for and been accepted to a program that they called the Day One Research Community, where I got to live in a dorm with all of the other first years in this program, and we would do a research course together, which would replace our first year of biology lab. And in this course, we got to work on a year-long project and present our work at a poster presentation at the end of the year. And in the first semester, each of us got to isolate and characterize our own mycobacteriophage. So we spent the first semester doing different wet lab techniques and, learning these techniques and how to do bench work. And in the second semester, we chose a few phages from the class that we then sequenced and got to work on annotating the genome of these phages. And this was such a great experience because it not only taught me a lot of different wet lab skills, but it introduced me to the concept of research as a career over the year. Because I guess I had known that researchers existed, but somehow it hadn't really clicked for me that that was something that I could pursue. So I guess this was realizing that there's a path that I can do research in a lab, I can get a PhD, and make my job being asking these questions about how the brain works and trying to find answers to the questions I had always had. So this really struck me as what I had been looking for. And for a variety of reasons, including that the small liberal arts program wasn't quite for me, um, I transferred to the University of Michigan, where I got more exposure to different types of research. Of course, I tried the typical lab courses, so chemistry, physics, and I ended up taking a research methods course for cognitive neuroscience. Um, and then I also tried out an independent research course where I got to work for a semester in the cognitive and affective neuropsychology lab at UMich, working with a grad student for course credit. And I was learning how to do human subject research for more psychology leaning experiments. And I found out that I was really interested in the big picture psychology questions, but I found that I missed the wet lab work that I had learned how to do first year. Um, and at the same time, I was taking a bunch of other different classes like genetics and biochemistry and was getting really interested in the more molecular basis of what's going on in our brains. So I was looking a bit more into molecular neuroscience, but I was a little bit limited in trying to look for a lab to join since I had spent college working throughout both the school year and the summer to be able to, um, pay for college, supplement my scholarships. So I looked through the job postings that the university had to see if there were any specifically paid research opportunities that I could take advantage of, so that I could try and commit as much time to research as I could while also meeting my financial needs. So this took me in a bit of an unexpected direction at the time, since I was offered a position in the Boyle lab, um, to act as a research assistant to help a PhD candidate. Um, and this lab focuses on identifying and investigating different regulatory elements in the human genome. So strangely, I ended up in a genetics lab, not a neuroscience lab, for the major research experience in my undergraduate education. But this ended up being super great because that research experience combined with my last semester, I took a course about neuroepigenetics. And between these two and a bunch of other opportunities, it really opened me up to the different areas of science that I was interested in. So then I graduated in 2020, and my last semester of college was cut short, obviously put online. And as you can see here, I stayed with my grandparents during the lockdown. So I got to graduate on Lake Huron, where I had grown up, which I guess is maybe a silver lining in that semester. But during my senior year, I'd kind of come into the realization that although I was pretty set on going into research and doing a PhD, I wasn't entirely sure what area of research I wanted to work in. I was becoming more and more interested in molecular biology and genetics, even though I was still interested in these bigger picture, more neuropsychology focused questions. So I wanted to explore a little bit. And I also wanted to have some time to save up some money prior to starting a PhD program. So I decided to apply for research assistant jobs, and this brought me to Massachusetts for the first time. And I ended up in Worcester working in the Flavahan lab at UMass Chan Medical School. So the lab, the Flavahan lab, the main focuses are one, trying to figure out the different epigenetic mechanisms that are involved in cancer, with a specific focus on glioblastoma, which is a type of brain cancer. And in addition, we also worked at improving different tools and methods to be used to study epigenetics. So towards that point, we worked a lot with the type of sequencing called nanopore sequencing. So as you can see in this figure below, nanopore sequencing, basically there's a flow cell where you put your sample into one end and it flows across a chip, which has a ton of little pores in it. And your DNA or your RNA sample flows across that surface, and each DNA strand is pulled through one of the pores. As it goes through the pore records the current as the nucleic acids are being pulled through that pore. And based on the current, the reading of how much current is at the pore at that given second, it is able to tell you which bases are in the pore at that point, and use this to be able to sequence your sample. But to use this technology to identify epigenetically modified bases, it requires creating a large training library so that the program can use that library as a reference. So one of the projects I worked on, which is the project for the paper that's in this lower right-hand corner, recently came out a few months ago. We focused on creating a method which enables researchers to create a training library to train this nanopore sequencing technology to read different epigenetically modified bases. So after about a year and a half of working in the Flavahan lab, I moved on to start my PhD at Brandeis, which is where I'm at now, working in the Griffith lab. And so my current research, I'll go into a little bit about. A lot of our lab focuses on this protein known as calcium calmodulin dependent protein kinase 2, or CAMK2, which is an important protein involved in synaptic plasticity, learning, and memory. It's highly abundant in in neurons, and the gene and the protein that it codes for are conserved cross species from invertebrates such as fruit flies and aplysia through vertebrates such as mice and in our own neurons. It's also known to be locally synthesized at synapses. So local protein synthesis is when mRNA are trafficked from the nucleus where they're made, as you can see in these little pink squiggles, throughout the cell body to distant parts of the neuron, such as the synaptic compartments. And there they can be translated at these specific synaptic sites when the protein is needed. So this allows neurons to make specific proteins at the precise time and place that they are needed. In our lab, we use fruit flies as our model organism, and I specifically look at the neuromuscular junction of the larva to research my questions, because we know that on both sides of this synapse, local synthesis of CAMK2 occurs in response to specific stimuli. So as you can see here, if we look at the synapse from the side in this cartoon that I've made, you see the presynaptic motor neuron in blue is synapsing onto the muscle, which has this postsynaptic specialization shown in orange. In both of these synaptic compartments, there's CAMK2 mRNA and ribosomes present. And in response to neuronal activity, these localized ribosomes in the synaptic compartments start translating the mRNA in the compartment. And this increases the amount of CAMK2 protein in each of these synaptic compartments. So my current research is figuring out how is this occurring and what mechanisms are involved in this phenomenon. So one avenue that I've been starting to investigate is whether ribonucleoprotein granules, or RNP granules, are involved in this regulation of CAMK2 mRNA. So RNP granules are these membraneless assemblies of RNA and various types of proteins that form due to the many different interactions that can occur between these components. And these assemblies can create biochemical environments which promote specific interactions between proteins and mRNA. And they can also be involved in many different types of mRNA regulation, including transporting mRNA throughout a cell, storing mRNA at a given location in a cell, and regulating its translation into protein at that location. And one of the things that makes flies so great to study is that they're very easy to do all sorts of different genetic manipulations. So one example that I use in my research are fly lines that have endogenously tagged fluorescent proteins. So I have some fly lines which have been genetically altered so that now in their genome, next to the protein that I'm interested in, there is a sequence that encodes fluorescent protein. This means that when the gene is transcribed into mRNA and that mRNA is translated into the protein, it results in the protein of interest, but with a fluorescent protein fused to it. And this allows me to use fluorescence microscopy to visualize this given protein. So, for example, there are a couple different proteins that are known to be found in these RNP granules I was telling you about. For this legend, I've just labeled them both as a granule marker with a little bit of detail below, but don't need to know the names of these specific proteins. In blue here, you can see the presynaptic motor neuron, which synapses onto the muscle. All of the magenta dots are one type of protein that can be found in some granules, and all of the green dots are another type of protein that can also be found in granules. So I can use these fluorescent proteins and the consequent imaging to look at where these proteins are that I'm interested in, where they're found in the cell, how much they're found, their different abundance throughout the cell. And in this case, when I'm looking at two together, I can see, are they found in proximity to each other? Do they seem to be overlapping, which suggests that they might be interacting? There's a lot of questions that I can answer just by using these endogenously tagged proteins. I'm also able to look at one of these proteins under a microscope that is continuously taking a series of images, which I can later stitch together into a video. So here I have taken a video of one of the endogenously tagged granule proteins over 5 minutes. And the warmer the color, the greater the fluorescence intensity. So these yellow orangish dots represent high fluorescence intensity of the tagged protein. If you see these two dots here, they seem to fuse together and let's see. They seem to fuse together around here as though they're coming together to interact. And then they seem to have some more movements and seem to, around 4 minutes and 20 seconds here, come back apart. So I'm able to observe the real-time movements of different proteins that I'm interested in. I'm also able to visualize mRNA using a different method called fluorescence in situ hybridization, or FISH. Here are two different experiments. So in this first one on the left, we see CAMK2 mRNA in green, and the nuclei of the muscles are in blue, and the presynaptic motor neuron is in magenta. On the right, we see an example of visualizing both a granule marker protein that I'm interested in, which is in green, along with CAMK2 mRNA in magenta. So with these different experiments and visualizing both CAMK2 mRNA along with proteins I'm interested in, I can see how does CAMK2 mRNA get from the nucleus to the synapse, visualizing and quantifying the location of these mRNA, the abundance in different areas of the cell. And I can use this to address questions about how different proteins are affecting the level and localization of CAMK2 mRNA. And I've included this slide that I end my department talk with because I think it's really important to note that in science, there's no such thing as science that's done just by one person. Not only do you need the support and knowledge of so many people collaborating together and learning from each other, because no one person knows everything. And by putting all this different deep expertise together, that's how you get great scientific discoveries. But also, community is so important because research is hard, a PhD is hard, and having a support system in your community is really essential, obviously in all aspects of life, but certainly working in scientific research.